for still being with us after a long day of presentations. I have the pleasure to welcome you to the PGM workshop. We try to make this nice and easy because all of you have seen a lot of slides today, so we'll not torture you with too many data. My name is Philipp Habermeier. I'm responsible for business development for our forensics business in the EMEA region, and it's my pleasure to show you what the Ion Torrent next generation sequencing platform can do for you in forensics. If you look at forensic DNA analysis today, I mean, this is something you're very familiar with. Currently, it's an STR-based analysis in most of the cases. With CE, capillary electrophoresis is regarded as the standard. This is cost-effective, fast turnaround, high throughput, gives you a profile in the majority of cases. And of course, it has helped to establish DNA databases based on STR profiles around the world. And you allow us a small commercial down there, okay, you can cover the full workflow with us. But of course, as we've learned today, we have the same game again now? Okay, there we go. The classical workflow I've just described with, I mean, you start with extraction, you might do quantification in case, for examples, run an STR, PCR, and then analyze on the capillary electrophoresis, of course, has its limitations. And in some cases, it might not give a full profile. And there are good reasons why also to look into NGS for forensics now. And some of these reasons are written on the slides. I mean, NGS will allow you to analyze more markers. So not only looking at STRs, you will also be able to analyze SNPs, mitochondrial DNA, also mRNA, as we've learned today from different talks. You have a capacity of higher multiplexing, so higher number of loci, but also different types of markers. Barcoding will also allow you to multiplex samples. And of course, NGS allows you to work with smaller DNA fragments, which will make it more likely to be helpful for analyzing degraded DNA. And then especially if you look at analyzing STRs with NGS, you will get more information, more accurate STR repeat counts. You will get more information about the repeat structure. And you'll be also able to detect allele subtypes, so in SNPs in the flanking region or different repeat motifs and the STR. What kind of applications are we targeting on the PGM? Well, some of them you have seen already. Analyzing degraded DNA. This is aiming of using these SNP panels we've heard about already, but also STRs, generate more investigative leads. This is looking then into using phenotypic information, ancestry information based on SNP panels, analyze complex familiar and kinship cases. Again, here we can use SNP panels, you can use Y-SNPs, you can use Y-STR information to analyze these kind of cases. Analyze missing persons or very much degraded remains. There, a mitochondrial analysis solution will be helpful. And there we are also looking in analyzing the full mitochondrial genome. And then, of course, mixture analysis. There are high hopes that NGS can help in analyzing mixtures much more efficiently, also again using SNPs and STRs to do this. If you look at the workflow, I mean CE workflow, this is something you're very familiar with. You start with your extracted DNA, perform an STR, PCR, do the capillary electrophoresis, and then you analyze the data. In NGS, it's similar. You also start from your extracted DNA, but here you need to first construct a sequencing library. So basically, this is targeting or using a targeted amplification for the targets you're interested in, for example, STRs. And then you have a library. You can use barcodes to use a multitude of samples. And then this sequencing library needs to be connected to a template. In the case of the PGM system, these are tiny little beads to allow the sequencing machine to do the sequencing. And then you do the massive parallel sequencing on the PGM system and you analyze the data. Okay. There we go. We are aiming to provide a complete workflow for HID, for human identification on the Ion Torrent system. You see, we have started with the first bunch of panels, the HID panels, based on our Ion AmpliSeq technology. For the template preparation, we are currently using the Ion Chef system or the One-Touch 2 system, and then run the sequence on the PGM sequencer. And then the Torrent Suite software is what we use to analyze the data, and there are different HID plugins for each application, STR, SNP, and Myto. And we heard some comments from, for example, from Runa in her talk, asking for automation of the library preparation. This is something we are hearing very loudly, and believe us, we are working on this. This is something that is coming. We expect it for the second half of the year, doing a library preparation also on the Ion Chef system. So there will be library preparation and template preparation both on the Ion Chef system later this year, which will of course help to reduce the time and also the number of manual steps in the workflow. Matt, can you do the next slide, please? Do it analog now. The technology seems not to be working. Okay, software tools. There is the Torrent Suite, and maybe to give you an analogy, the Torrent Suite is maybe something like, you know, from capillary electrophoresis, the data collection software. Torrent Suite basically allows you to plan your runs, and it's also doing a basic data analysis, detecting your SNPs, detecting the variants, and then in the next step, Matt. And then we have what we call the HID plugins, so they are additional level of analysis software that are then, of course, aimed at the forensic analysis. And we currently see next generation sequencing really as a complement to the existing capillary electrophoresis workflow in situation where, for example, a quantifier trio kit might tell you, okay, there's a degradation going on, or you only get a partial STR profile or no match in the DB database, then you might want to start looking at your sample also with an NGS. Why it does make sense to analyze degraded DNA, we try to show you with this graph. So you see these purple little diamonds. These are the data points for an STR kit analyzed on a CE platform. And you see on the y-axis we have the probability of identity, and on the x-axis, the further you go to the right, the more DNA is degraded and the less loci you recover. And you see, for example, in this situation, in the middle, where it says 7 loci on the STR data, the STR kit is only able to recover 7 loci, but you get much, much more information still, so about 53 SNPs from the SNP panel, and you see that the discrimination power you still get from the SNP data in this example. So I think it's very promising also for identity. What we have seen some data from testing these panels, the identity and the ancestry panel. These are ready to order kits with 96 reactions, and we also have specific library kits for them. If you want to try them out, they are already available. Next one. Yeah. Just a few examples on the data analysis. This is very visual. You see some analysis here, for example, on the Y markers included in the identity panel. You get a random match probability calculation from these samples. And there's also a profile comparison tool in the software. And Matt will, after my talk, give a live software demo, so because that I will not talk too much about the data here. Ancestry panel information, you see, I mean, basically, what you see in the lower half in this map, on the world map, you see sort of a heat map overlay. This is basically a graphical representation of the likelihood that your sample might originate from this population. Currently, we have to be very careful. I mean, with the number of SNPs we have in the panel, it gives you a very rough estimation on broad global groups. And of course, this can only be good as the number or the population databases this are relying upon. And if you want to have more granularity on a biogeographic level, of course, we would need to include much more SNPs for a specific population. But as the first start into this direction, you see it's providing some interesting data already. Yeah, SDR analysis, we are currently in early access, which means we are providing kits or panels to early access test site labs. On the upper left half of the image, you see a CE picture, fragments, and below you see NGS. So this is a shift in the mindset of you analyze the data, millions of reads for a specific region. Then, of course, what you will see is a lot of more information, for example, on the repeat structure on STRs, like in the example here on the D2. So you see the allele 20 in this example. There are a lot of different repeats that will occur in NGS. Yeah, this is, again, you see just an example on STR allele subtypes, sequence variants detected by NGS for this given loci. How does it look like? I mean, this is just an early version of our STR analysis software, but you see something you are very familiar with. You see an allele table in the upper half. Then you see, very similar to GeneMapper IDX, you see a plot of your loci, and then you see the alleles. In this case, for the D3, a 15 and a 20. And if you would take the mouse, hover over, you would see the number of reads for each allele, and then in the lower part you see it goes down to the sequence. So you really see the sequence information on your STR for each allele. Yeah, Niels was so kind to allow us to show this example we've seen before. See a comparison of a sample. On the left-hand side, there seems some heavy degradation going on. Only the small markers are recovered by the CE, and we have a nice full profile on the PGM system, which is a promising result for a first prototype they've been using in this example. Yeah, and this is already also available, published on FSI Genetics, if you want to look at the details. Next thing that is coming up as a panel from our side is a Myto tiling pass panel, spanning the entire human mitochondrial genome in two primer pools. You can see the details here. And it's also based on AmpliSeq technology, and it's supposed to be available in the second quarter. Yeah, we think capillary electrophoresis and next-gen are a powerful combination for forensics. Just some considerations in terms of throughput. You see on a CE platform, currently we do up to 27 STR markers. This can be very fast depending on your workflow. If you have a direct amplification kit, for example, for STRs, you can get a profile in a few hours. And it can be comparably low in cost. In contrast, on the PGM system, you can sequence hundreds of markers at a time, different markers, so STRs and SNPs. It's more suitable for degraded DNA, and also NGS on this system, it's a very fast method. So if you take just the workflow alone, with all the preparation manual steps, you can get from sample to result in 24 hours. But of course, no lab is working 24 hours around the clock, so if you do a normal 8-hour work shift, it might be 2 or 3 days, depending how do you set up the workflow. And from the application area, I mean, CE, it's clear you do STR fragment analysis. SNPs have been analyzed, but smaller multiplexes. MitoDNA has also been done on CE, but mostly the control region. And in contrast, now on NGS, on the PGM, you do a sequencing-based STR analysis. You can do larger SNP assays, and of course the complete mitogenome, and then of course also looking into mRNA, tissue, body fluid ID. These are also applications that will be possible. Yeah, with that, we think we have an integrated forensic workflow that allows you to address your standard samples, reference samples, but also difficult and specialized applications together with both platforms. And I think what is maybe important to highlight, we will more be happy to help you with guiding you the way from CE to integrating NGS in your workflow. And we also have the bioinformatic experts that can help you get up to speed in how to analyze this kind of data. We are also organizing something we call forensic schools. These are specific training courses we offer on the PGM system. We've done 3 of these courses last year with the help of Walter Parson and Peter Schneider. Small groups, 6 to 8 people in our training facilities, in our labs for 3 or 4 days. And it's, I think, a very good opportunity to learn the workflow, get your hands on the instrument and learn how to work with the new technology. There will be new dates announced soon on our website. This is the link to our human identification training website in Europe. Please check back, and we will of course also communicate by emails as soon as the new courses are available. And with that, I want to thank you for attention and hand over to Matt for the live software demo. I think because we have some technical issues connecting Matt's laptop here, you will hear him talking from behind and seeing the software screenshots up here. I hope you're happy with that, and I will just stand by if in case there are any comments needed from my side. Thank you.
Okay, thank you, Philip, and hello everyone. I'm hiding over in the corner in case you can't see me. I'm over here not only to advance Philip's slides for him, but also so I can operate my computer and show you the some detail of what PGM data actually looks like. So simply just to give a bit more detail and to make it seem more real after all the presentations we've had today, what I'm going to do is just walk you through what PGM data actually looks like for a person in the lab. So to do that, I will close out of the presentation we were just looking at and come over to my browser. And this is not only how I'm going to access the data remotely now. This is also exactly what the user of a PGM would see in their laboratory. When you run a PGM, you generate a really large amount of data with every run. And this is managed by a server, we call it the Torrent server, that sits next to your instrument and collects and handles all of this large amount of sequencing data. And rather than the way it is on a CE machine, where you then generate your data and then transfer that data to the computer that you wish to analyze on. The way that you'll typically interface with your sequencing data on the PGM is that the data will stay on the server attached to your instrument, and then you will remotely connect to that using any other computer of your choosing. All you need is a web browser. Now this, of course, it can be set up so that you can access your data remotely through the internet. This is what I'm going to do now. I'm going to connect my web browser to some of the torrent servers that we have in the company and show you some PGM data. But if that's a concern to you in a forensic laboratory, then don't worry. Then you don't have to connect your system to the internet. You can simply just use your web browser to point on your internal network in your laboratory, and the data never leaves your premises. So, however you achieve it, you open the web browser like I have on the screen in front of me, and then you enter the address of your server. It might be very small, but you can maybe see it up the top there. And then you're presented with a login screen, and it looks just like a web page, and you log in. Save a little bit of time though. What I've done is I've already logged in, and I've got an example of what's called a run report in front of me. And this is something that you'll generate for every run that you do on the PGM. And it's really the first interaction that you have with the data coming off your sequencer. It's a way that the data is summarized and presented to the user so that they can begin to make sense of these really large amounts of sequencing data that the instrument is producing. So before you get into your application-specific analysis and thinking about your STRs or your mitochondrial DNA or whatever the case may be, the run report is designed to first of all, just simply give you an overview of the run in question. And you can evaluate the quality of it, see if you've simply sequenced some DNA. So the first few metrics that we see on the screen there, the most prominent one is in the left-hand side there. This map, we call it the heat map, and you'll see that it's measuring what it refers to as ISP density. So this is your loading. This is how well you have loaded that chip. If you're familiar with the way that we do the sequencing, and if you've been out to our booth out in the foyer, you can have a look at the PGM itself and have a look at some real PGM chips. But what you do in the lab is that you load your DNA into that semiconductor chip. The chip goes into the instrument, and then we try to detect DNA in every tiny little well on that chip. So when we've done a run, what this map here is showing us is simply, first of all, did we manage to get some DNA into that chip, into each of the wells on the chip? And it's color-coded, simply such that red means that there is a lot of ISPs in the wells. Yellow was fewer, but still a decent amount, and then down to green and blue few and almost no ISPs respectively. If you're familiar with the chip, that has that characteristic oval shape across the middle, so it's only this area here where there are wells to be loaded. The corners will always be blue, indicating that there are no ISPs here. So what the system will give you is a percentage score of how many wells you've managed to get an ISP. ISP is the name of the bead that we attach the DNA to. So it's telling you if you've done a good job of loading your chip. And you'll see it gives you a little score out of 5 here. In the example I've got on the screen, there's 80% loading, which is pretty good. And you'll see that the map is mostly red and yellow. Okay, so moving on then, our next question is, well, we've managed to load some ISPs into the chip. What does that mean in terms of DNA sequence? How much sequencing have we done? And so this is where we can look to the middle of the screen. And these bars tell you what wells, how many wells you managed to get ISPs into, and then as you go further down, it tells you whether or not you had good quality DNA in them. So the run in question is of a 318 chip, which has about 12 million wells on it. And you can see here at the top, this first metric is the same one that we saw here, the 80% loading. So 80% of our wells were loaded, and that means that we got just over 9 million wells. So that number might be a little bit too small for you to read there, but this number here in the middle of the blue bar is just over 9 million. So the next question is then, of those 9 million wells that we got an ISP into, how many had useful DNA for us? If we look further down, the next score is what's called enrichment. So that means, did those ISPs have DNA attached to them? And in this case, the enrichment is typically a very efficient process, and 100% of our ISPs had DNA attached. So again, the number here is still over 9 million. We look a little bit further down. The next one refers to what's called clonal and polyclonal fragments. So you could consider that if this one measures how many of our beads had too little DNA, this is talking about how many had too much DNA attached. And so it's a little bit beyond the scope of what we're trying to achieve here to go right into an explanation of what the difference between monoclonal and polyclonal is. But for these purposes, it's enough that a polyclonal read is one that has too much DNA attached to the ISP, and the system cannot be sure of the sequence in that well because there are different types of DNA sequence in there. In this case, and this is a fairly typical score, we had 70% clonal wells or reads, which is the desired result, and 30% that were polyclonal. The instrument and the system, the software, will automatically filter those out for us. Moving on to the last one, there's a few final filters that occur. Again, the text might be a little too small to read, but just various types of low quality and control fragment are taken out. And in the end, we're left with 5.9 million reads that reflect our final sequenced library. So this is our desired and final result. It's DNA that we have sequenced that is of sufficient quality to go on and make further analysis with. And then this is the number reflected at the top here, just over 5.9 million reads, 5.9 million wells on the chip where we have got usable DNA. Okay, so moving on then. We look over here and we start to see some statistics on the read length that we have achieved. We have a histogram of how many reads we managed to sequence plotted by the read length that we achieved. So this, of course, will be characteristic of the multiplex that you've used. If you've made amplicons, then you will expect to see a collection of jagged bars that reflect the size of the amplicons that you've made in the initial library preparation. And then there are some statistics here of, on average, how long the reads were that we achieved. And then taking those two together, you'll see a statistic up here of the total number of bases that we've sequenced. And in this case, we've done 587 million bases worth of sequencing. Okay, so that's a brief introduction to the way you start to interface with your data when you're working with the PGM. And as I say, this is a single page report. It's generated for every run you do, and we refer to it as the run report. You can export it as a PDF and save it into your quality system. And as we look further down, there are some other metrics about accuracy and alignment and so on that I won't have time to go into in full here, but they will let you do various things like, for example, for most runs, for most HID applications, you'll have multiple samples that have got different barcodes all pulled together and being sequenced on the one PGM run, as well as looking at the overall sequencing for your chip. You can look at how the sequencing performed on a barcode per barcode basis. So you can see here that for the 10 barcodes that we used in this run, we achieved this number of reads for each of them. And then if you look further down, there are various other options, and we can see a histogram for each of the individual barcodes. You can see if any one sample performed well or performed poorly at this stage. And then you also have some options to download files with your data. You see you have access here to some file types, including the BAM file type that you can use to put into other software programs if that's what you'd like to do. And even further down, we have access to FastQ files. So again, if you've got third-party applications that will take a FastQ file, you can take those and import those into the other software programs. So that's the run report. And as I say, it's the first window that you have into your PGM data. Moving on though, once you've done this analysis and you've concluded that you've got some high quality sequence coming off your run, well, the next thing that you want to do is to see what that means in terms of your application. You want to look at the STRs, you want to look at the SNPs, and whatever that might be. You want to analyze those profiles, and this is where the plugins that Philip mentioned come into play. So depending on your application, there'll be links in your run report that will take you directly for a given run to the plugin result. I've got them open here already in other tabs just to save time on the loading. This one here is an example of what you see in our HID SNP genotyping plugin. And we've seen throughout the day some examples of what the data coming off this looks like, but this is just a chance to see it live, if you like, in front of you, see exactly what the person in the lab might see. If I scroll up to the top, you'll see some just summary information about the run. Again, you have the chance to download some files that give the details of what's in your run. You can make a PDF report of the plugin result. But moving further down, we start to see the application-specific SNP data that we achieved in this run. Down the left-hand side, it's a little bit compressed on the screen because I'm connected to the projector. But we have a list of the barcodes in the run in question again, and you can see that I'm clicked here on the top one. So barcode number 7 has been selected, and then when we look to the middle, we can see some information specifically about the barcode that you have selected. So in this case, This is a run that's been done with our ancestry panel, and you can see it's a little bit faint in some places, but you can see the world map here, and the likely ancestry of the person in question is highlighted. You can see Southeast Asia there is highlighted, and then if I change tabs, it'll give you that same information in tabular form. So this is the table that will tell you the likely admixture proportions of the sample in question. In this case, the software has concluded that the person in question isn't of mixed heritage likely, that they're likely to be of 100% East Asian ancestry. And then, again, we saw some examples of this during the day. The software will also attempt to put some confidence bounds on that estimation, and in this case, tells you that it's high. The final tab of the three is simply just, you could consider it the profile, so just a table of all of the SNP loci that have been analysed, and then the genotype in question for each of them for this person. So this is the box here. You'll see that I've just closed, called the admixture prediction. If I move down to the next one, it gives you some statistics that are simply another way of looking at the same data. In this case, it goes through, and the additional information it gives you is a random match probability that's been calculated from each of the ethnic groups that are stored in the database. We have a default database that is supplied with the plugin that gives you some allele frequencies, but if you have allele frequencies of your own, you can program those into the plugin, and it will use those for you. The important point to make with this table is not so much the number that you get for any particular ethnic group, but the striking thing is the way that the geographic regions will tend to cluster, and you can see that East Asia is almost all of the top categories here, again indicating that this person is likely of East Asian heritage. Moving further down in the plugin, you start to see some more metrics that let you evaluate your profile. So up the top, it was a case of what have we done with this profile, and it showed you the SNP genotype. Here you can dive into it into a little bit more detail, and there's a table here that has one row for each locus in the multiplex, and as I say, it's become a little bit compressed because I'm connected to the projector, but for this first row, for example, it's telling me that the SNP in question is on chromosome 1. There's the RS number of it, and then in this sequencing that we've just done, we achieved 550 coverage. None of those were A reads for the given SNP. 260 were C reads, no G reads, and then 290 T reads. And then if you look a little bit further down, that's concluded logically enough that at that locus the person in question is the CT genotype. And of course, similar to gene mapper software, if you like, there are some settings behind this analysis that you can customize that will define some of those cutoffs, as in how much coverage do we need for the call to be made, what should the balance between the two different alleles in a heterozygote be before it can be called. You'll see some other metrics here, forward and reverse reads, in this case 207 and in one direction and 343 in the other. You can set some bounds on what those should be for the genotype to be called in the settings of the plugin. Other than this, you'll see here I have two tabs, and then if I change to the other one, it gives you similar data but in a graphical form. Across the bottom, you have all of the loci in the multiplex, all 165 in this case, but of course it's a little bit too small to see the detail of that many. So what you can do is you can simply drag a rectangle, and that will zoom in the screen above to highlight whatever you've selected. And so the graph shows you a bar for each locus. and then the vertical axis is the coverage that you've achieved. And then the color coding tells you the base that was observed for each of the SNPs. So the ones that are a solid color, and I can hover my mouse over them, you'll see there's the TT genotype. The neighboring one that's split into two is the CT genotype. And I can do some things like I can sort this by coverage. You can see here from highest to lowest coverage at the end. And I can normalize the display, which is if you want to examine in closer detail the balance of the two alleles, that can be a useful option. And then again, you can export the data, either a copy of the picture of the chart or download the table if you'd like to put that into another program or Excel even. Okay, so that's a brief overview of what you see and what you can do with the SNP plugin. I'll move on and show you the STR plugin. So in a similar way, you would evaluate your run report for an STR run. The same considerations of looking at the heat map and looking at the enrichment and looking at the number of polyclonals still apply. It's just the case that when you come to do the application-specific analysis, rather than using the SNP plugin, you use the STR plugin. And so this is a live version of what we saw in Philip's presentation. And again, it's become a little bit compressed on the projector, but at the top we have a table, and so there's one row for every sample in my run, and it's a table that looks very similar to the type of table that you get out of GeneMapper for a CE run. And then if you highlight any given row, then the charts below will populate, and then you can see the data and the charts for that sample. The default view is comparable to what you'll see in a CE run, in that you have coverage, which here, which is your strength of signal, if you like, rather than RFU. And then across the bottom here, you have the size of your allele, as in the number of repeat units inside it. And so these views will be look very similar to those that you'll see in a CE run. You can even see little bits of stutter here on the left-hand side of the taller peaks. And then if you hover your mouse over a bar, you can see that this one here represents the 10 allele, and this one here the 14. But the next thing that you can do that essentially illustrates the power that you have when you're actually sequencing the STR data is that if you then click onto any of those bars, the view will shift, and it will show you what sequence represents the bar that you just clicked on. So in that case, I clicked on the 10 allele here, and you'll see the view shifts to a green bar, and that 10 allele is specifically made up of the sequence that you can see there. In this case, there's only one type of sequence, but we've seen some examples throughout the day that what can appear to be just one allele in a CE-based view, when you're just looking at the number of repeats and that's all, if you actually look at the sequence in that allele, it can actually be more than one allele, and it could indicate that you had a heterozygote when you thought maybe it was only a homozygote, or it could tell you some information about a mixed sample that would make resolution easier than you could have with CE. And then to complete the picture, if we look further down, we have that same information in tabular form. So I have a nice illustration here selected already. You'll see here in sample number 13, you see the apparent 15 homozygote selected here for the D3 locus. When we look into that into more detail, you'll see that there are actually two different alleles represented, even though to a size-based method of separation, it only looks like one, because they both have 15 STR repeats inside them. You can see the difference there. It might be a little bit hard to see, but there's just a single base difference there in A to a G. And then those two variants, there's 1450 of one and 1361 of the other. So some strong evidence there that we have in this sample a heterozygote rather than what looks like a homozygote. The final thing I'll show you then is that both in the STR and also the SNP plugin, there are links that will let you easily show your data in IGV. And again, IGV was mentioned several times during the day. It's a third-party program that will let you view your sequencing data, and I've got it open in the background just here for this one. And so just a nice link that will automatically take you to the locus in question and actually let you visualize your sequencing data. And you can see here for the locus in question. If you're not familiar with IGV, you'll see the bases across the bottom here represent the reference, and then these bars up the top here represent our reads that we achieved in the sequencing run. Now the different colors reflect the different directions of sequencing, so forward and reverse, and you'll see maybe the most striking factor is that almost all of them are missing a 4-base stretch in here, but this is simply that the specific person that we have sequenced has got one fewer STR repeats than the reference. You can see an example here of one read that's missing 2 4-base stretches next to each other. That would be one of our stutters. It's a fragment that is one entire STR repeat shorter than the expected allele. And then to illustrate the point, you can see scattered down some of the reads here what is an A in the reference, and about half of the reads is represented here as a G. So this is our variant sequence that lets us tell the two alleles apart. Okay, and with that then, we've had a trip through what data looks like coming off the PGM from the run report and then into the sequence specific and application specific analysis that you can make later. Okay, so with that, I'm finished, and thank you. So back to you, Philip.
Thank you, Matt. Okay, thanks for staying so late with us. That's all we had prepared for today for the workshop. I hope it was useful. If you have more questions, come and see us at the booth. We'll be there tomorrow or later on during the dinner. Please come if you have more questions. Thank you for staying. Thanks.